unfolding is deadly. Okay? Alzheimer's is obviously another disease where you have this kind of unfolding that takes place. Okay? Again, proteins denature, and they tend to influence other proteins to be denatured to beta sheets, and hence that causes the entire misfolding. So this is nothing wrong genetically, at least to our knowledge right now. And you have the protein, it's a perfectly functional protein, the sequence is perfect, but somehow you're getting a broken protein after that protein is perfectly translated into a functional protein. Just denatures, and whatever causes the denaturing, we don't know. But that's one of the reasons of the mad cow disease. By the way, Sprengel is also mad cow disease. Again, it's eating basically those proteins, and they used to feed the cows, or the cattle in general, some other the cows for protein sources, and so they end up having those prions in high concentration, and they will eat them, and that will actually travel to the brain and cause this problem. That's why now we have grass-fed cows rather than the old way of feeding cows other sources of proteins. So anyway, it's a problematic thing. So misfolding protein can be deadly. And this is just to show the kinetics of folding. Protein usually is in some sort of equilibrium between folded and unfolded, and this sigmoidal curve basically represents how the folding to unfolding events happen, to where it's very slow until you reach a threshold of protein that are unfolded, and then you get a cascade and a very fast transformation to unfolding. Same thing happens in the opposite way as well. This sigmoidal curve, by the way, because we will see it later, maybe this week or next week, when we talk about binding in hemoglobin. Now, Leventhal paradox. This is just some fun exercise that Leventhal did back in the, I would say, 50s or 60s, where he's trying to study the mechanics and the mathematics of the folding. And he did this interesting experiment. It's a thought experiment, computational experiment, where he had a protein, a fictional protein, okay, that the amino acids in this protein has 3 conformations. Okay? You just assume that each amino acid has a maximum of 3 conformations, which is, of course, an underestimation. But at least for this exercise, it's good. And so if you have 100 amino acid protein, then it will have 3 to the power of 100 possible conformations. Okay? Now remember, in our bodies, in any living system, folding, as soon as the protein translated, takes less than a nanosecond, probably much less. It's immediately almost ready to function. So he's trying to find out the timeline of this based on his calculations. And so he found out that it will take that many seconds in order for the protein to fold properly based on this experimentation, because he assumed a nanosecond per right conformation, and that will take that many centuries for each protein, if that were the case. Obviously, this is not the case. So that means the folding doesn't happen one amino acid at a time. Not every amino acid has to find its conformation and then do it. The folding probably happens in a very localized and fast way. Okay. This is actually part of the explanation of how protein folding happens. If you look at this statement here, we think it's like a weasel, and you give the monkey a typewriter, and have him type this in the hope that the monkey at one point will construct a sentence by just randomly typing, any way the monkey can type. And so you will never actually reach the stage where the monkey will be able to produce this sentence in this kind of coherence. However, if you have in that computer a program that will immediately lock the letter that is in the right place. The monkey is just typing, and once the letter in the right place appears, the computer will simply lock it by combination. Then you're going to have a progression where you can really, within our lifetime, the monkey can type it, just with the aid of the computer. So in the proteins, at least we believe, that you have a folded protein, an unfolded protein, and then quickly the local structures, the adjacent amino acids, will be able to find their secondary structures immediately. Like the demonstration I showed you on the website yesterday, where you have local folding. Once the local foldings are done, then you can have the protein structure. So it's really sort of like a cone. Things happen very quickly after you get the localized folding. All right. So that brings us to proteome studies. Obviously, proteins are the workhorse. Really, this is what we use in order to diagnose a lot of diseases. Without them, many bad things can happen. So proteomics is basically a fancy way to say that we're studying multiple proteins in a system. That system might be an organism, a group of organisms, or etc. So what happened here, from the genomic era, the DNA sequencing era that started in 1995, we ended up having an incredible amount of genes that we had no idea they existed. And hence, from the genes, we were able to get these incredible number of proteins. And so proteomics, of course, concerns itself with studying the function of those proteins, including finding the structure of those proteins. Okay? So that, we're going to dedicate this lecture actually to some of the techniques that are used in the lab in order to study different aspects of proteins. Now, this is really a brief, way to study this. We're just talking about it briefly, and I will tell you what you need to know just for the class. But otherwise, we're going to try to cover some of the aspects. We can tell a lot from those techniques that we've been developing for the last 150 years. And they are really simple, quite effective. So the first one I want to talk about is centrifugation. That's basically a centrifuge. You put stuff in, it spins very fast, and it can separate stuff based on the density and the size. Okay? Now, that's what I want you to know about centrifugation. We can prepare stuff based on centrifugation. We can separate proteins, we can separate degrees. You can see here that if you go 10,000 times the gravity for 20 minutes, you'll separate a certain part of the cells, certain fractions. If you go 100,000 times for 1 hour, you'll separate other organelles, and so on and so forth. So, centrifugation is really one of the first things you use when you study any kind of system. Okay? Now, salting out, that's a very interesting technique. That's basically adding some sort of salt, and ammonium sulfate is one of the most popular salts. This is to separate protein, to unsolubilize it. When a protein is soluble, we want to get it out of the solution without harming it. Usually we use salting out. And depending on the size of the protein, we can add different fractions of ammonium sulfate to precipitate different fractions of proteins. And this is what people usually do in order to separate their protein during purification. And just to show you how it works, let's say this green ovals, those green ovals are proteins that are soluble. If you will see that the surface amino acids are usually the charged amino acids, some of the polar amino acids, and they are in relation of water, between these different hydrogen bonds, right? And that keeps the protein soluble. So when you add salt, okay, you can see those salt here, ions, they will interact with water. Then they're smaller and more agile, they will have priority. So they will tend to take water molecules out of protein. Now, depending on the size of the protein, you might not precipitate it immediately just because the protein is in a competitive size. But as more salt, eventually they will start interacting with the water and taking that water away from the protein. What's going to happen is it will simply separate out. Hence, will aggregate, and when the water is going to aggregate and precipitate. And then you can put it in the centrifuge and get your protein. Right? Now, in this aggregated form, the protein is not active. It's unfolded. It's a mess. So what we do is we do something called dialysis. Dialysis is that bag, that nylon bag, that has controlled sized pores in it. Usually you put this mess of protein and ammonium sulfate in it, and we just put it in a buffer, as you see, and that tends to take the things out, the salt out, because it goes from higher concentration to lower concentration. Right? So you can see here the protein in red, the salt, let's say the blue dots, and you put it in a buffer that has no salt, and so things will go always from higher to lower concentration, and they will empty, and you will have eventually a protein with much less solution, with much less salt. If that's not enough, then you will take this bag and put it in another buffer that doesn't have salt in order to purify it. But the ultimate result is that you're getting this protein that doesn't have it. Okay? And that protein usually comes back functional and folded and ready to go. So it's a non-aggressive way. Salting out. This is what it's called. Salting out. Okay? With ammonium sulfate. All right. Another way out is gel filtration or chromatography. Chromatography is basically a matrix, okay, that you put in a column, and then you put your protein there on top, okay, or your protein mixture on top. And then gravity takes control of things because it's going to have to go down this matrix. As it goes down this matrix, proteins will separate based on different sizes. The smallest one will move faster, and the largest one will move slower, and you will be able to collect those fractions. It's a way to isolate proteins. My itself is basically made of spheres of some sort of carbohydrate derivatives. If you cut this out, you will see that inside this there is this mesh that has the average control size. The proteins will be traveling through. Smaller proteins will travel slower because they will go through all these meshes, while larger proteins will be running over this and run faster. So I said smaller protein will run faster. No, a smaller protein will run much slower because they have to go through all these things. Okay? This is called chromatography or gel filtration. So if you're running that with a salt, the salt will be much, much slower because it has to go through all these, and proteins will run much faster. So this is another way of salting, getting rid of the salt. It's called chromatography. Okay, this is what it looks like. Of course, those are much smaller than this. And basically, you just put the protein. This is what the gel looks like. And you can see it's basically this white matrix. You will come with the protein mixture and very carefully lay it here, and then start collecting droplets from here, and you keep adding the buffer. And your protein will travel. Now, how to find out that protein, if it doesn't have a color, that's different. You don't have to worry about. What I need you to know from this, chromatography or gel filtration, separate protein based on size, to where smaller size proteins will run slow and larger size protein will run fast. Okay? So it separates based on size only. Okay? So does the folding of protein play a role in it? You bet. I'll give you an example. Here I brought these 2 sheets of paper. If you have a protein that looks like this, and you have a protein that looks like this, which one is going to go faster? This one, the compact one. So really when we talk about size, we talk about effective size. Especially if we're running native, in other words, the proteins are their size. Those will run slower than those, because those are very small, compact. It's the same size of amino acids, but it has to go through those little holes in those gels. This will roll because it's very big. Okay? So when we talk about size in chromatography, we mean the effective size, whether the protein is denatured or not. Okay. Another flavor of chromatography is ion exchange chromatography. This is the gel filtration. This is basically shows you how the bigger proteins will run faster than smaller proteins. But that's really what I want you to know about. Okay? We are not going to talk about how we will get the fractions. Really subject for the next class, next course. Ion exchange chromatography. It's the same kind of beads that I just showed you, except they have a charge on them. They have a charge, either positive charge or negative charge. And hence, the proteins will stick to those based on the counter charge. Okay, that's called ion exchange chromatography. And so the separation is based on charge. Size has nothing to do with it. Only the charge of the protein makes a difference here. So if we know our protein is negatively charged, then we can use something that exchange negative charges. Okay? And hence our protein will stick there. An example, so we have 2 kinds, obviously. An anion exchanger that means it will attract negatively charged proteins. A cation exchanger will attract positively charged proteins. And obviously this is a very important feature. If you know something about your charge of the protein, the charge of your protein, then you know which one to use. Okay? Of course, the size of the protein matters as far as the number of charges on it. If the protein is more charged than the others, it will stick higher. Then there are ways to get rid of that protein afterwards. That's the different story. So ion exchange chromatography has cation and anion exchange chromatography, depending on what we're exchanging, and the separation having based on size. The jet filtration chromatography separates based on size. Okay? Those are the points that we need to know. All right. Now, this is just peculiar stuff I really don't want you to know, but I want you to envision what's happening in ion exchange chromatography. You can see here the matrix. You can see this green thing is a protein that's negatively charged. It's going through, it's positively charged, it's going to attach to it. Okay? Now, the way to get this protein data to unstick from it, is by competition, by adding something that's also negatively charged, that's smaller than the protein, and that will simply compete and will block this, and the protein will fall off. Okay? Another thing that we are very familiar with is by changing the pH. We've seen how changing the pH changes the charge of the protein, right? We actually were calculating that. So if a protein is negatively charged at the acidic pH, we increase the pH, we make it neutral or positively charged, and that will allow us to leave the column, unstick from it. Okay? And hence, it's very important to be able to manipulate these parameters in order to get the protein you want. Now, the last flavor of chromatography is called affinity chromatography. And this separation is actually the best of both, because it depends on the specificity of the protein binding to a chemical. Anything. The example here, so those are the same kind of beads, the matrix we talked about, except they're modified to cross-link with that specific chemical, that bait that our proteins love. For example, let's say our protein, the one in yellow here, loves to bind glucose. Okay? And so we have those beads, we can make them, we can add glucose, modify them. So now we have our bait stuck permanently to those beads. Now all the proteins will go on, because none of them like glucose, except our protein. And hence our protein will simply bind to it very specifically. And hence the affinity. Everybody else will go on. Now if you want to elute means get our protein back from the column, because it's stuck there, then we can add free glucose, just like we added ions in the ion exchange. The free glucose will come, the protein will see the free glucose in an overwhelming concentration, and it will prefer to bind it. Of course, that free glucose is not stuck to the column, and hence they will both fall, or elute, and hence we will get it. Okay? That's called affinity. That's actually the best way. If you really know what your protein likes to bind, then you will get it almost immediately. Yeah? And in the biochemical methods class, whoever is going to continue on this, you'll actually know the details of this and how we can genetically engineer our protein in order to bind to specific targets. So this has been, there's been incredible advances. Another technique is called electrophoresis. Now we're going to cover the electrophoresis again when we talk about nucleotides. Electrophoresis is basically running a protein in an electrical current. Okay? If your protein is positively charged, it will run towards the negative pole. If your protein is positively charged, it will run to the negative pole. If your protein is not charged, it's not going to run at all. Now, SDS, PAGE stands for polyacrylamide gel electrophoresis. Polyacrylamide is the matrix that makes those gels. So it's also a mesh, and the size of the protein matters. Except in this case, smaller proteins will run faster than bigger proteins. So it's the opposite of chromatography. Now, it's called SDS because we use something called SDS, which stands for sodium dodecyl sulfate. Sodium dodecyl sulfate, it's a very simple structure. It looks like this. That is sodium dodecyl sulfate. Usually it has sodium here, as an ion, as a salt. All right, what do we notice about sodium dodecyl sulfate? It's a very long molecule. So is it hydrophobic or hydrophobic? So it's interesting about this, because when we add this to a protein, this long thing acts like a crowbar that goes inside the hydrophobic inside of the protein. And it disrupts, just like urea and guanidine chloride, disrupt those weak bonds, those hydrogen bonds and ionic interactions. And that causes the protein to unfold. Not only that, because it's negatively charged, what's going to happen is that protein effectively will be coated with negative charge. So now we have an open protein that is negatively charged, despite what is the original charge of the protein, because this will overwhelm the protein. So now imagine we have different proteins, thousands of different proteins, they're all exposed to this. It doesn't matter how they fold it, it doesn't matter what their relative sizes, they will all be separated on size and only size, because the charge to size ratio will be the same for all of them. And hence, when you denature them this way, and you have them all negatively charged, and you're running through a current, they will all run to the positive pole, first of all, and they will all be separating based on their size. Okay? Now, sometimes we can also dissolve this with dialysis and get them out and get the protein back, but sometimes not. Usually it's used as a prognosis to look for certain proteins that we absolutely know what is the size of that protein. The nice thing about this is that, so this is the acrylamide. You don't have to know this. Okay? This is not important. It's important, but it's not important for the class. But this is how we polymerize it and make those meshes. This is your sodium verutyl sulfate, and basically is 1.4 gram of sodium verutyl sulfate per gram of polypeptide. So the mass to charge ratio is constant, and hence we separate on size. Now, what about a protein that have internal covalent bonds, like disulfide rings? Would the SDS affect them? The SDS has nothing to do with it. So the protein will remain partly bent if there is SDS. And usually, if we want to get rid of that, we have to add something like beta-methactylethanol. Right? We know that already, that beta-methactylethanol destroys those disulfide rings. All right. So you have your protein, you put this in, and eventually your protein will look like this. Right? They all will look like this, with same kind of proportions of negative charges around it, and then they will run here. Right? So usually, actually always, we run with our protein sample. Our protein sample will be loaded here. It will be run on lane number 2. Okay? And this is supposedly our protein, this band here. Always we run on number 1, what we call molecular weight markers. Molecular weight markers. Those are proteins that we absolutely know what their size is. Why? Because we want to estimate the size of our protein. So our protein, just looking at this here, it is between 45,000 and 31,000. We know that. If we know that, then we know that this is our protein. Make sense? My question? Now they're all subjected to SDS, and they're all running from negative to positive because they're negatively charged, and they're running based on size. The smaller protein will run faster, as you can see here, and the larger protein will run the slowest. And our protein will run accordingly because it has the same mass to charge ratio. Yeah, please do interrupt me if you have any questions. So what we can do here, we can actually have a better estimate of the size of our protein because everything runs linearly, hence it's a very nice estimate. Say it again? Yes, that's a very good question. What about different proteins? It doesn't matter what protein you have, it will all be negatively charged because SDS will basically destroy that tertiary structure. Unless it has, again, the disulfide rings. Then you're going to have to add methactylethanol. But you don't know. So sometimes we add methactylethanol anyway, just to make sure that to level the thing. All right. So this is a very good time for math. So because they are linear, the distance from here to here, we can actually correlate that distance with the size. So if we measure all those distances and we correlate them to the size, we can have a very a plot that looks like this. This. There you go. This is our ruler, and this is our plot. And you can see here, those are our known standards, and we can actually make a linear regression and have a nice straight line if everything runs perfect. Now we measure our protein. We measure this with our protein, and we just simply put the distance here, and then we can know the size of the protein. It's pretty actually accurate. It's not 100% accurate, but within 100 kb, it's fine. It will definitely tell you if that's your protein or not. Okay? So you can estimate the size very easily because of that linearity, because everybody has the same size to part ratio, and hence everybody runs the same distance. All right. Any question about this? Okay, so this is SDS page. That's what I want you to know about SDS page. It has SDS in it, that denature the protein based on disruption of the secondary of the weak bonds, weak interactions, not the disulfide group. And hence the proteins will have equal mass to charge ratio, and they will run equally on the gel. Smallest proteins will run faster than larger proteins. Okay? Smaller, faster than larger. That's opposite to that gel filtration. And because of that, we can actually estimate the size. Now this stain here, we call this coma C blue. There are so many other stains, because we cannot see the proteins really. And the coma C blue tends to actually aggregate and around proteins and give us this nice blue color that we can see. So we have to stain the gel. But that's a procedure. There are so many stains. Okay. All right, moving on. That's another electrophoresis. This is called isoelectric focus. This is so cool. This one is very cool because that depends on the PI, the isoelectric point of the protein. The trick here is to have a gel or matrix that is gradient in pH, that has pH gradient. That basically it has different zones, and those different zones have different pHs to where you go from low pH to high pH. So now the protein is traveling in different pH environment. So as we've seen yesterday, the pH affects the overall charge of the protein. So we load the protein somewhere in the middle rather than here or here because we have no idea. We do not need to nature the protein. That's the power of this procedure, that the protein shouldn't be denatured. We want the protein to be in its native structure and hence its native charge. So we load it in the middle and we run the current. Now, depending on the pH of the middle, let's say 7, the proteins will have different charges and hence they will be moving to different zones. Positively charged proteins will move towards the high pH. Negatively charged proteins will be moving towards low pH. And they will keep traveling in this continuously changing pH environment until they reach a pH where the total charge will be 0, which is its isoelectric point. What's going to happen to the protein at that point? It will stop. No matter how much electric current you have there, it will stop. And then you take that gel and you develop it by adding some sort of stain, and you will see that your protein is somewhere. Yeah? Make sense? This is called isoelectric focusing. That's what I needed to know about isoelectric focusing. It's a separation, it's an electrophoresis that separates proteins based on charge. Not size. Not based on charge. Based on PI. Based on the isoelectric point of the protein. Based on the pH of the protein where the charge equals to 0. All right. ELISA. ELISA stands for enzyme-linked immunosorbent assay. Now, this is a very specific diagnosis. This actually is used a lot in home kits, like pregnancy tests and stuff like that, because it's very, very exact. Because it depends on something we call antibodies. Antibodies are part of our natural immune system. They're produced by the body to neutralize any kind of invaders, bacterial or otherwise. Okay? Those are very specific. They have memory. They can bind only one thing and they can bind it very good and neutralize it. We can make those actually in the lab, and we can make them against certain proteins. So if we have those made against certain proteins, or against our protein, then we can simply use it to only target our protein, and only our protein with 100% accuracy. So ELISA, we have usually this antibody that has an enzyme linked to it, enzyme that does some sort of color reaction. Here's the enzyme as E, and here's the binding side of the antibiotic. The protein will basically go through this kind of liquid, right? And it has our antibody. So what happens, the antibody is right here, then we add our protein, and if it colors it, if our protein binds, it will make the color change. Okay? That's all I need to know. It's basically binding based on antibody. That is very specific, and it gives the color. And that from that pregnancy test, it will give yes or no, because it either will bind that protein, that particular protein that's related to pregnancy, or it will not bind it. And hence it will give that color, or it will not give that color. Of course, people can say yes or no, but that's all based on the same. Finally, Western blot. Western blot is actually invented by a guy called Western, and hence it's Western blot. And here, we run the protein on a SDS electrophoresis, and then we take that and we blot it. Blot it means we take that matrix and we put it on a paper in order to transfer the proteins from the gel to the paper. We can then take that paper and we can add a radioactive antibody. Okay? And this here represents a radioactive solution that has our antibody, and then we can take it, or it has some sort of fluorescence, and if our protein exists, then we will see it. If it doesn't exist, then we will not see anything here. Sort of like ELISA, except on a sheet of paper. So both Western blot and ELISA, the one thing in common between them, they use antibodies. In the Western blot, we use electrophoresis to separate the proteins and get the antibodies. That's all I want you to know about it. Yeah? I talk a lot, but really not a lot we need to know. You just need to know the concept of those things. And when do we use these things? Without any paying attention to detail. All right. So hence, let's see what do you think of this next question. What is the function of SDS and SDS electrophoresis? Okay, what is the best technique to filter out a small contaminant, such as salt, from a solution? Okay, now based on what we talked about. All right, let's talk about the last technique. It's very fun. It's protein sequencing. Okay, I'll tell you what we need to know about it. But basically, let me tell you what protein sequencing is. Protein sequencing is basically knowing that amino acid structure of that protein, knowing the primary structure of a protein. That's what protein sequencing is. Okay? Knowing what are the amino acids. Now, granted, this is not as popular as it used to be before the genomic era, the sequencing, the DNA sequencing era, where they used to sequence DNA, and now we can know the sequence of the protein because of the translation, which is something we'll cover later next week. But sometimes we really are looking for protein. The DNA doesn't tell us exactly if that protein is expressed or not, which is something I'll be talking about as well. But we need to sequence that protein. So we will do the SDS, gel electrophoresis, and then we can actually, with an X-Acto knife, we can get that band out and get that protein out. Okay? And then we can take that and sequence it. Know what is the sequence. We probably cannot sequence the entire, let's say, 100 amino acids or 200 or 300. But we can sequence reliably up to 40, 50 amino acids. And that will be enough to tell us what that protein is. Okay? So how do we sequence a protein? How can we read the sequence of a protein from a sample? Okay? This is, by the way, one of the old sequences. It hasn't changed much, by the way, in principle. But Edmond is the scientist that actually invented this method that is still in use. It's called Edmond degradation, and the name, of course, implies that you are degrading your protein, you're destroying your protein. Excuse me. You're destroying your protein one amino acid at a time. Okay, and in this destructional process, you're doing this bad thing to the protein, you are reading it one amino acid at a time. All right, so here is the technique in a nutshell. This is a protein, a peptide. Okay? You can see here amino acid 1, 2, 3, 4. And of course, in between them, you can see those peptide bonds, right? You can see those peptide bonds. And the thing that we are adding here is phenyl isothiocyanate. Okay? PTH. Let's go to PTH. Okay? I need you to know this particular chemical, because that chemical can only bind to the N-terminal. It cannot bind to the C-terminal. It cannot do anything to the protein. It can only bind to the first protein, the first protein because, again, we are always talking from N to C, from amino terminal to the C terminal, the double. So it binds here. Because of this binding, is that now this peptide bond happens to be weaker, because now you have a resonance of the electron that extends all the way to the PTH. Okay? So that means if I would take this now PTH protein and treat it with a mild acid, this will simply break off. You will have the PTH derivative of the first amino acid. And you will have here your protein minus its first amino acid. Now the question is, how can we know what is this? Then, PTH, we know that it's PTH, right? But we need to know what amino acid is attached to PTH. Well, what we can do, one of the many things we can do, is we can put it in chromatography. Size chromatography, gel filtration chromatography. Now it's based on size, not just protein size, any chemical size. And because we know the sizes of all 20 amino acids, we know that very well, by the way. We actually documented. We can know exactly each amino acid, how long does it take to travel through this column, how many minutes. Yeah? And we can detect that. So we can put it in, and then we can see that it traveled within that many seconds or whatever, and then we can detect it. Okay? And then we can take the next amino acid, we can do the cycling again, add another PTH, and clip the next amino acid from the internal and run it through the chromatography and run it and see what's the next amino acid. So effectively what we're doing, we're reading the amino acid one at a time from the N to the C. Isn't that amazing? You can read it from N to C. Okay? Just like you clip it. And you get the sequence. That's it. Now, there are far more sophisticated techniques now. Actually, there's spectrometric techniques where you're actually using the mass of the entire peptide. We don't have to worry about this. It's called Maltitov. You'll find it in your book, actually, in chapter 3. We're covering chapter 2 and 3 today. And, you know, if you want to read about it, it's very fantastic. Okay, but this is how this protein. This is enzyme degradation. It's usually good for about 50 amino acids. That's what I want to know. Plus the PTH and how we do it. And that's it. That's really it. Okay? Now, another way, we have tools, very, very effective tools, that can break specific peptide bonds. Those called peptidases. Remember? We talked about them very early on. Protein that destroy or reagent that destroy peptide bonds in a very, very specific manner. Always predictable manner. Those are molecular tools, actually, we can use to manipulate proteins. Those things in red boxes here, you see those? There are three things here in boxes. I want you to know those. I want you to know what they destroy, what kind of peptide bond they destroy. So, for example, reagent-wise, we have cyanogen bromide. That always cleaves the carboxyl side of any methionine residue. It always does that. Okay? So when you break a protein and you run it in jet electrophoresis and you see two segments there, you know that you have one methionine in your protein. Right? Because cyanogen bromide only breaks methionine. Right? Trypsin, it's a very famous protein because tryptic, we put protein with trypsin in order to break it, and they always break the carboxyl side again, the carboxyl side. Remember that the amino acid has the carboxyl and N side, the amino side. So it breaks the carboxyl side of lysine and arginine, and only lysine and arginine in the carboxyl side. So if your protein has no lysine or arginine, it will never be broken by this protein. If it has one lysine or one arginine, then you will have two segments, and so on and so forth. But you know, right there at the junction, there will be either lysine or arginine in your protein. Chymotrypsin, the carboxyl side of tyrosine, tryptophan, phenylalanine, leucine, and methionine. Dear God. Well, that's going to be hard to remember. Well, but what's in common between all those amino acids? They're all hydrophobic, right? As a matter of fact, three of them are aromatic. Tyrosine, tryptophan, and phenylalanine. Those are the three aromatic amino acids. So you can say the aromatic amino acids that automatically tell me tyrosine, tryptophan, and phenylalanine, and then leucine and methionine. Those both are also hydrophobic amino acids. And it breaks only at the carboxyl side Again, they come out this side. Okay? Now, there's one here that I don't want you to memorize, but carboxylyltretase is possibly the only one here that breaks on the amino side of things. Okay? So I need you to know those three. What does come from this? Well, I can give you a sequence, and I tell you that I treated it with trypsin. What is the result? Okay? And I'll give you these choices. Okay. Which type of protein purification relies on attraction of protein to a specific or particular chemical group? What kind of those techniques work on this? Let's see. Well, that should be a simple one. There's one technique that only relies on the specific binding of that protein to certain things. Okay, so what we studied today, in conclusion, dialysis, chromatography, just to kind of use it to organize your thoughts, all right? Infiltration, ion exchange, affinity. We didn't talk about XPLC, we talked about isoelectric focusing. All right, here's isoelectric focusing, those are the three electrophoresis, and ELISA and the Western blot. Now, this is actually just to show you how, you don't have to worry about it at all actually, but if we have a protein of unknown sequence that is here in red, okay, we don't know the sequence, and let's say we treated it with trypsin, and we got those two fragments. We sequence them and we see those two. Now, how can we put them together? This is called assembly. And basically, you put them together this way, and what happens is you can then deduce, basically, and this is a very simple way to do it, really. Deduce the size of the protein based on that. And so you can put them together and you can know the things of the protein. But this is basically how people use it. Of course, on bigger fragments, it's difficult, and now with spectrophotometric ways, it's much easier. All right, thank you very much. I'll see you tomorrow. Yeah.